Yes.
Okay, perfect. All right. Can we see my screen?
Yes, we can see your screen.
Okay, perfect. Okay, yes, so I'm Ford Combs. I am a senior bioinformatics engineer at Biotia. And today I want to talk a little bit about GeoSeeq and how you can use it for metagenomic sequence analysis, particularly as it pertains to public health. And for the first part, I'm just going to spend like 10 minutes or so to talk about the background of this kind of approach, how it can be used, the types of things that can be done with it. And there may be some echoes of what you heard yesterday from Dr. Neve O'Hara, who's the CEO of Biotia. Maybe some of you were at her presentation yesterday. If so, I hope that you enjoyed it, and I hope that this is a good add-on to that. So let's get started. So we can start by thinking about metagenomics as an approach by looking at the microbiome, right? So a microbiome is a community of organisms that can coexist in a particular environment. There can be hundreds of different organisms in these kinds of environments, and they can change over time. So maybe one of the most well-known microbiomes would be the gut microbiome. And there's some interesting connections there to health in general between the types of bacteria that live in your gut and the axis between the gut and the brain, and the ways that your lifestyle and your diet can influence the microbiome and change what's going on in your mind and your health in general. So there's some pretty interesting stuff going on there. And on the right side of the screen, I have a figure from a really cool paper on sourdough starter microbiome. So sourdoughs are basically combinations of flour and water that are left to ferment. And the fermentation is caused by a community of bacteria and yeast. And at the top part of this figure, you can see different colored bars, and each column corresponds to a sample, the sample being one sourdough starter from someplace in the United States, and I actually believe around the world as well. And then the bottom part is the type of yeast that's present, and the top part is the type of bacteria that's present. So by doing metagenomic sequencing, you can actually see the different organisms that are present in each of these starters, and you can see how it varies based on where the sample was taken. So moving on here, when doing these kind of characterizations of these microbial communities, one of the common approaches is to do 16S sequencing, which I think many of you are familiar with. So you target the 16S gene, and then you amplify that gene and you sequence it. And by comparing the sequences of this variable region to known taxonomic sequences, you can then determine what organisms are present in a sample. Typically, you can go down to the species. Sometimes you can only go down to the genus level. And then you can also look at the relative abundance of these organisms in your sample. And this will be on the left side of the figure, on the right side of the screen. The metagenomic sequencing is quite different from this 16S approach, in that instead of targeting a specific portion of a gene, you actually randomly target the entire genome of all the organisms that are present in the sample. And so by doing this, you're able to actually look at different types of genes and functions that might be occurring in a particular organism. And we can kind of see this a little bit on the right in like the functional analysis that's mentioned at the bottom. You can also potentially get sequencing coverage for AMR genes that might be present in some of these organisms. So you can do some AMR profiling. And you can often get down to subspecies level, so strain level identification of the organisms that are present. Another huge important difference would be the cross-domain coverage. So if you're only targeting the bacterial 16S gene, you're not going to get information on fungi that might be present in your sample. And metagenomics is domain agnostic. So if there is a fungus living in that sample, its DNA is going to be just as sequenceable as the bacteria might be. So with the metagenomic sequencing data, there are several things that can be done downstream for analysis. One of the first types of analyses that is typically performed is some kind of taxonomic classification. So typically you have a read file. This is called a FASTQ file. And if anybody has questions about this, please don't hesitate to stop me and I can give a little bit more detail about this. So a FASTQ file is really just a file that contains nucleotide sequences and associated quality scores. When you're trying to classify these sequences, one of the common approaches is to use a k-mer analysis. So a k-mer is simply a substring of a larger string of length k. On the bottom here, I have a little toy example. So this is a little sequence, and I've broken it down into the first 3 k-mers. You could imagine continuing this for the rest of the sequence. And then I have a little table here that shows the number of each type of k-mer, or at least the first 2. So we see that ATG only appears once in this sequence, whereas TGA actually appears twice. Now, a k-mer of length 3, it doesn't tell you much about a nucleotide sequence, but when you extend that k-mer length out to something like 31, for example, you actually get to a level where k-mers can be unique to individual species. And that's the point where these different classification tools can come in. So you can take a set of reads, and by looking at the k-mer content of these reads, you can see where those k-mers map, and each taxon where those k-mers map, it kind of can be thought of as evidence. And the more evidence you have for each read mapping to a specific taxon, that allows you to classify that read. And on the right, we see like a kind of a breakdown you might see from this type of analysis where different taxa are present at different levels in the read set. And we'll see more about this a little bit later when we look at the actual GeoSeeq application. Another important analysis that can be performed on metagenomic sequencing data would be searching for AMR genes. And I mentioned this earlier a little bit, but the basic idea here would be to either do a read mapping approach or an assembly-based approach. If you do a read mapping approach, what you start with is a database of AMR gene sequences with known phenotypes. So a particular AMR gene like the, I don't know, BLA-OXA gene, and you might say that that particular sequence is linked to a particular type of resistance. So then what you do is you map the reads that you generated during sequencing, and if you get enough reads mapping to a given gene in your database, that's an indication that that gene is present in the in that metagenomic sample, and therefore the organism that it is associated with may be there and may be resistant as the phenotype shows in the database. The other approach would be an assembly approach, and the assembly approach is going to come up a little bit later when we look at an assembly, but in this approach you actually start by taking metagenomic sequencing reads and you assemble them. And when you make an assembly, what you're trying to do is find overlaps in the reads, and by finding these overlaps in the sequences, you can actually build out longer sequences. So even if your read length is only of length 150, if you start to find regions of overlap, then you can build out long, what are called contigs, that could be really any length up to a certain extent for the number of reads present in your data set. And this is something called an assembly. Once you have this assembly, you can kind of use that same database in the other direction. So instead of mapping reads to the database, you actually take the database genes and you map them back to your assembly. And so if you find an AMR gene in your assembly, then there is a very strong chance that that gene is present in the sample, because of course the assembly is derived from that sample. And so these kinds of characterizations can be done in metagenomic analyses, and we see an example of that on the right here, where we have a number of samples taken from all over the world, and you can look at the different AMR genes that are present on the bar graphs here on the bottom of the screen. We can see the different types of genes that were found. Okay, so I want to touch briefly on machine learning because it's really important, especially in the context of GeoSeeq. So when you have these metagenomic data sets, we have a given sample with some number of features associated with it. These could be taxonomic classification features. These could be AMR genes that were present. But we can take that initial set of features and we can extend it. One of the data sources that we can use GeoSeeq for would be looking at climate data, for example, or the geographic distribution of the samples. We also have rich metadata that can be combined to that sample. Once we have this large feature set, we can start to think about machine learning. So in machine learning, you could have a set of samples with known classifications, and then you have this feature set for each sample. You feed that into a model, and this is kind of what we see a toy version of on the right here. And that model learns to associate those features with a given class. And once the model learns this process sufficiently, you can apply it to samples with unknown class, and then have a good idea of what the class of that sample is. So building these types of models, especially using data from all around the world and collaborating with different people, you're able to build really extensive models that have the potential to, say, track emerging pathogens and hopefully prevent future pandemics. I want to touch briefly on this thing called UMAP because we're going to look at some graphs later. And I think it'll be helpful to see this. I just mentioned for the machine learning, we can take these data sets with lots and lots of features and train models. But oftentimes it's very useful to reduce the number of features to a smaller amount so that you can, one, visualize what's going on like we see on the right. I'll explain in a second. And also we can have a more accurate machine learning model because of the way that the data is condensed. What the plot on the right shows here is each point is a sample, and it's colored based on the climate, the Köppen climate classification of the source of that sample. And the position on this chart is the principal component, is actually called PC1 here. This is the component number 1 on the x-axis and component number 2 on the y-axis. So this high dimensional data set of taxonomic features has been reduced to only 2 features, and we can see the layout of the samples in this chart. And really, this is just a visualization example, but like I mentioned, you can still train models with these reduced data. So just to wrap this up, we've seen that metagenomics allows us to get cross-domain data from a microbial community sample. We can do all sorts of analyses on this data, such as taxonomic classification, AMR profiling, and machine learning. And all of this type of work can be accomplished on the GeoSeeq platform, which I'm going to jump to now. So this is the GeoSeeq landing page. This is the main dashboard. And again, the idea here is that we want to be able to easily access data, upload, download, organize it, share it, and run analyses on it. And we try to do make all those things possible on GeoSeeq. So what we see on the left side here is options to look at different organizations and projects, which we'll look more at in a minute. We can also see down here, and this is really important, this documentation will lead us to just what it sounds like, GeoSeeq documentation, which can teach you all the things that I'm about to I'm going to explain part of these, but this will give you a more in-depth understanding of the actual application. So we can see some of the map features here, which I'll be talking about, and some of the different apps that are possible to be used here. So the documentation is really important. It's right down here in the support dropdown. So back to the dashboard, we can see the total number of organizations, projects, users, and the current number of samples on GeoSeeq. We can also see how that number has changed over the year 2023, and the most active projects. And then there's this really important map pane here, which is really cool. So I'm going to expand it. We can actually see different samples and where they came from across the globe. And we can change some filters on this background to get a better idea about where these samples may have come from. So we could look at the temperature. We can also look at precipitation. And you can pick samples based on, this kind of background that you're seeing. Maybe you want to look at samples that came from regions with high precipitation, for example. You might want to look at something like malaria as well. And for that, I'm actually going to go back to the base layer and pick malaria. And here we can see some hotspots for malaria. We could zoom in down here, for example. And so this is a great way to visualize some of this data and where it's landing on the globe. We can also look down here. If you just kind of move your mouse down to the bottom of the map, this bar this bar plot comes up that shows the collection date of different samples that are being displayed on this map. And so right now, this goes back to 2000, but we could change this to say, go back to, let's say 2015. And if we change it to 2015, you can see that the numbers have changed up here and the values have changed on this bar chart as well. It's actually a very small window. So I'm going to change this to 2023. And yeah, so we can see the different kinds of samples that are available for the different collection dates.
So for
Is there a question?
no, actually not really a question, but
Yeah.
some of us actually haven't actually looked at the GeoSeeq yet. So I would like to actually tell people that if you just to Google it, GeoSeeq, then you can go to this website. Am I right, Paul? Right?
Yeah, for sure.
Yes.
That's right.
So please
Yeah,
go
so
to
please
the
check it out.
website. Yes. Okay. Thanks.
Great, thank you for that.
Yeah, go ahead.
Okay, so yeah, let's actually pick some samples here. So I'm gonna go over here to Korea and pick out Seoul here. So we can see we have 63 samples showing up on the map. And if I click show samples, we go to this sample page. So these are the 63 samples that were shown on the map. We could go back to map view here and we can see them. But in the table view, we have each sample, and then the columns are the different metadata columns. And there are a few that I just want to highlight. So for example, we have the public transportation line. Oh, it sounds like there might be I think somebody might need to mute.
Oh, hold on a second. Yes. Oh, so okay.
We good?
I think so. Yes.
Yeah, okay. Great. So yeah, so we can see some of the different public transportation lines here, the surface material. So there's some great metadata associated with the samples. Let's pick a sample and take a little deeper look here. So this sample is part of the cell paper project of the Metasub Consortium organization. And we'll look a little bit about organizations and projects more in a second, but just focusing on the information we have here, we can see that there are several different folders. And these are the kinds of folders, the kinds of outputs that you would typically see when you run any kind of metagenomic sequencing and analysis. So a very common thing to see would be like clean reads. So clean reads would be FASTQ files, which we see here. And these are read 1 and read 2. So these would be paired end reads. And clean reads just means that either contamination or other sources of noise have been removed. So this could be like removing human reads if you were maybe taking a gut microbiome sample or something like that. But it's removing unwanted genetic material from the read set. So this would be an example of a type of file that we would typically see. We can also go down here, the FASTQ, or FastQC rather. FastQC is a super commonly used piece of software. It's typically one of the first steps in any kind of genomic analysis pipeline where you actually run the reads through FastQC. It will remove adapters and low quality reads and perform trimming. And it also produces some really great output files for you. They are in an HTML format. So what we can do here is actually click on this file. And this preview is very small, but you can see that this is the FastQC report. We could select it and download this file if we wanted to. In this case, I'm just going to click this preview because it's a little bit bigger and we can see some of the data here. So for example, this is a FastQC chart of per base sequence quality. So what we're seeing is on the x-axis, each position from like position 1 to 150. And on the y-axis, we're seeing the quality score, the Fred quality score. So 30 would be 99.9% like quality score, so very, very likely correct. And then as you see along the positions of this read, there's a drop off as you get towards the end, and that's super common. We see that all the time in sequencing. Just to highlight another plot on here, for example, there's this per base sequence content plot. So here we can see the percentage of the different nucleotides down the length of the read and how it varies across the read. And what FastQC does is it gives you some kind of basic gut check quality ideas here, right? So we have these checks and these X's, which are an indication of potential problems in the data. It's a very quick and dirty report, but it's very useful for an initial analysis. So just because this has an X here, that doesn't mean that it's like a bad read set. It's just something to flag your attention. Going back here, I also wanted to show the GeoSeeq search folder. So we have a couple different files in here, and these are the kinds of files that can be created using different apps on GeoSeeq. And we'll look at some of the apps in a minute and actually run one. But here we can look at, for example, the taxa sunburst. Actually, let me go to this preview. So here we can see the taxonomic abundances in this great interactive sunburst plot. We can see that the human abundance is around 20%, and the Cutibacterium acnes is around 9%. And it is interactive. So if you just wanted to look at bacteria, you could select bacteria, or you could select the genus. You can zoom all the way back out, which is really nice and really convenient. Okay, so that's an example of some of the types of data that you can get when you do metagenomic sequencing and analysis here, but the organization of this data is also really important to note. So I already said that we have this sample as part of this project. Let's go back and take a look at this project. So here we see the project page. We have a few different tiles on this dashboard, and I'm just going to highlight the UMAP one for a moment here. So I mentioned this earlier. This is a way to reduce the dimensions of a data set. In this case, we're looking at the first and second components from a UMAP dimensionality reduction. And each point is a sample, and it is colored by the continent of origin. And the position on this plot is a it's the reduced dimensions of the first and second components of this taxonomic distances between these samples here. And what we can see is that if we look at East Asia, for example, it tends to be on the left side of this plot. If we compare that with, say, North America, we seem to have a lot of the points towards the right or in the middle. And then for Europe, kind of similar to North America. And you might expect some split like this. If you were taking samples from around the world, you would expect samples that are closer to each other in space to perhaps also be closer in the taxonomic distance. And that's exactly what we see. Another great thing about the project pages is we can have this documentation, which is really useful. So you can come in here and read information about this project and understand better about what's going on. And I'll actually show you how to do this in a moment here. And then back to, let's see here. So now we can actually take a step back further. And now we're in the Metasub Consortium organization. We were just in the Cell Paper project. And we can see some different projects here. So for example, this one, we can look at the dashboard here. Again, we have a UMAP plot. Here we have that kind of taxonomic sunburst plot we saw earlier, and we can see one of these for each of the samples in this particular project, which is really nice. We can zoom in to whatever level we want. Very useful. Here's another plot which we haven't seen yet. So this actually shows the percentage of reads that are mapped to a given classification group. So these different groups down here. So unclassified viruses, eukaryota, bacteria, and archaea. And we can see that the number of reads mapped to each class changes per sample, which is not surprising. And you can see the kind of distribution of these classifications vary quite a bit across the samples. And a little bit further down, we have this bar chart which shows the species abundance in the project. So we see Homo sapiens, for example. We change this to prevalence. We see it a little bit differently, the different species that are here. And we could even change this to genus. So a lot of different ways to look at this data, to interrogate this data. One more project to briefly show. Where is it? It's down here. Yeah, searcher demo. Yeah, this is just another dashboard I wanted to show and highlight. It's got similar functionality to the other ones we've seen. And I think what's really cool about this is you can run these different kinds of apps, get these different kind of analyses, and have this great dashboard page for your sample set. And then you can also have this documentation, which is super useful. So this really aids in collaboration of different groups that are accessing the same data and trying to run different types of analyses. Okay, so the last segment I want to do with this is actually create a project. So I just want to show you how to actually do this. I created an organization earlier. You could create an organization if you'd like. I created one called Example Org 2. So if I click on this, I don't have any projects yet, but I can just make one. Pretty simple. I can go here and say Example Proj 2, and I can create it. It'll take a moment. And that's it. So now I have a new project and there's nothing in here yet. But let's say I want to add some samples to my project. Well, we already started by going here and picking samples off the map, but maybe we want to take a different approach. Maybe we want to, I don't know, maybe we want to search for samples from London. So we could search London. And so here we can see a large number of samples that matched the query. We can see where they are on the map, and this should unsurprisingly be, yeah, in London. So that's good. And then you could check out this data and see if it's what you would like to include in your project. And just for the sake of the example, I'm just going to choose a few samples here and go down here to manage samples. And I can add these samples to an existing project. So I can pick example org 2, and then there's my project, add those samples. And then here I am back in my organization and project, and there are my samples. If I wanted to run some analysis, I could go over to the apps page here, and I could add an app to this project. So here are the different ones, some different ones that are available at the moment. I'm just gonna pick this one, can add it, and then I can run it, and I can select the samples I want. Maybe I want to select all of them. Maybe I just want to run this one. And then there we go. So now it's running. If I go to runs, it should pop up. Let me refresh. And there we go. We see that the app is now running on this particular sample. I can also go over here and create some documentation pretty easily. So I can name this page introduction, for example, and I can make a little header that says, this is an example project. And I can say, new samples London to this project and create that page. And now anybody who's a member of this organization can come in here and click the documentation and learn about my little project. Okay, I think that's all I wanted to do with this for now. Also, I want to make sure there's time for any questions. So I'm going to really quickly go over this little Jupyter Notebook example. Let me go back to this real quick. So if we go to the support and documentation, you can see here that there is this tab called API/CLI. So the CLI would be the command line interface, and the API would allow us to connect with GeoSeeq from the command line, or programmatically, basically. And so in this, what we're looking at here is a Jupyter Notebook, which is basically an environment to run Python code. I think my understanding is that most of the people on this call have minimal exposure to Python, maybe a little bit. And I just basically, all I want to demonstrate here is that we can access GeoSeeq programmatically, and this is really useful if you're trying to either upload a lot of data or download a lot of data, run a lot of analyses. Being able to do this programmatically can really aid in making that efficient. So here we can see an example of running a search. So this would be like the term that I ran earlier. So London, you could also run Staphylococcus if you wanted to search for samples that were from London that had Staphylococcus in them. So if we run this, we will get the results that we would get on the GeoSeeq application, but now they're back in our little Python environment here. So here would be the results. We can see the city is London for all of these. We can go a little further and drop any of the rows where the latitude and longitude is what's called NA, or it's not applicable in this case. It has no value that's useful to us, so we can drop those rows. And then we can come down here, and using some basic plotting functions, we can actually plot these samples on a map. So that's pretty cool. That's a programmatic way of accessing this data. I also wanted to show briefly, because a lot of this kind of work, this kind of bioinformatics work is done from the command line. There is a GeoSeeq package that you can install with Python, with pip rather. And all I did was type in GeoSeeq here, and we're getting a little help message. I have that help message over here as well. We can see the different kinds of commands we can use. This is in the case of using the download argument, but if I go here and just type GeoSeeq help, these are the commands. So we can run any number of different commands. For example, download, which I just had here. And we see that in this module, we have different commands we can run to do different things, download specific files. And each one of these will have a help message. So if we run it to download files, this is the help message. It's a little more extensive because it shows you how to do specific things. I'm going to pass over this example and just get to one that I think is maybe a little more straightforward. So this command right here is saying download files from the Metasub Consortium Cell Paper project. So let's go find this real quick. So we can go to Organizations, Metasub Consortium Cell Paper. And then I want to look for this sample. So I'm going to highlight it. So it'll make my life a little bit easier here, and copy that. And then I go back to the project and click on this samples tab, and then I can search for it. So there's my sample. I'm going to click on that, and I could download any number of files from that correspond to this sample, but I want to look at one in particular because I mentioned it earlier. So here there is an assembly graph.fastg. So this is a type of file that's output from an assembly step. And to pull that file down, I indicate that the folder is a sample, and then I indicate that the extension is a .fastg. And again, a lot of this information is present in the help messages and in the documentation, and we're going to continue to make efforts to make this easy to understand and use. But in this case, we're just going to run this command. I've already done it, so I'm not going to run it again. And what we end up with over here is that file. So I've downloaded that file from GeoSeeq right here. And now I have it on my computer, and I can work with it. I've gone ahead and printed out some of the first lines from that file here. Maybe you are familiar with a FASTA file. This looks a lot like a FASTA file. It's slightly different, but we basically have a header and then a sequence. And I have an example of what an assembly looks like. It's kind of the last thing I wanted to show today. So this is what's called a bandage plot. I'm not sure if anybody has seen this before, but what this is trying to show is that when you make an assembly, a de novo assembly from reads, you are trying to find overlaps in those reads to build out these, what are called contigs, these larger sequences which we can fit the reads together and form. What this plot shows is that these longer blocks are longer contigs, longer sequences that we were able to create using the reads from our sample. You can see there's also a lot of segments with these short little blocks. And if we zoom in on those, actually, I can't really zoom in very well here. Well, I guess I can do this. If we look at some of these sections, we see a lot of little blocks, and you can see multiple lines connecting different blocks. And what that actually means is that you can't resolve that sequence. Typically means there are repeats in the sequences. And so there are multiple ways that the reads could overlap and connect different segments. But this is the kind of structure you might expect to see when you run reads through an assembly pipeline. And so with that, I will stop. And if anybody has any questions or wants to see anything else again, please let me know.
Okay. Thank you it was really informative. Before probably people have many questions, but
Okay.
I would like to ask you a few questions first.
Sure.
So the last part, the assembly part.
Yeah.
If I understood correctly, then since we have our samples are metagenome sequencing. So in my mind, if we try to do assembled, then I'm expecting have many of assembled ones, right? Because there are many different species in the sample.
Yes. And yes, that is true. This would be an assembly. This is just an example. And I believe this is from this is from an isolate sample, I believe. So this is one species. And even in that case, you can see there are a couple of little offshoots that couldn't be connected.
Right.
In a typical metagenomics assembly, though, you're going to see lots of little pieces like this, basically. So you see lots of smaller ones. Now, there are ways of dealing with that. So if you first do a taxonomic classification and you see that you have, say, Staphylococcus aureus, maybe that's one of the species in your sample, then you can try to pull out the reads that belong to Staph aureus, and then you can assemble those. And then you might end up with a nice, cleaner assembly, right? Or you could map your reads to a reference genome of Staphylococcus aureus. If you just assemble all of the reads as they are, you can end up with lots of different kind of really, I guess, kind of contigs that are connected in different ways. You could end up with chimeric contigs where different species are connected together because they happen to share a very similar sequence. So it can get pretty messy when you're doing it from the kind of larger metagenomics approach. But there are ways to facilitate doing assembly for sure.
Okay, so basically, with the shotgun metagenome sequencing data, assembly, just the simple applying assembly this code may not really good idea. Maybe we should pick the one species and then try to look at whether we can assemble that species whole genome. Is it the way we should go?
Yeah, I think they're different approaches. I think there are different reasons you might do an assembly. Like if you want to do an assembly to have a genome of a specific species, that's one kind of approach, right? If you want to do an assembly to look for, say, AMR genes, then you might not necessarily need to do any kind of major work to separate things apart. You might just do an assembly and then look at those genes map. And then some of those genes are going to be species specific, right? And then you don't really need to worry about the full assembly to try to make a genome. Rather, you're using it as a tool to create highly covered regions of genomes that are present in the read set. So there are different reasons you might do it. Does that make sense?
Yes. So thank you. Now I understand that. Oh, the other question that I have is that actually this part is quite important for us. So the ones you explained to us is based on the the sequencing data already present in GeoSeeq.
Mhm.
How about we have a new sequencing data? We have a Pasteur files.
Yeah,
Can we upload them into the GeoSeeq and then run the analysis? Is it possible?
absolutely. Yeah, absolutely. That's the idea, right? So you can go to upload samples, and you can pick your organization and your project, and then you can select your files. So you could do bulk upload, and you can just drag and drop your FASTQ files right here. You could upload a single sample as well that way, and then you would have your FASTQ files on GeoSeeq.
Okay, I see. In that case, after we upload the pasta file, then
Mhm.
we need to have the clean version, right? To look at the downstream analysis for us.
Right.
So, how can we create those files, clean ones, or some
Yeah.
of
So
the files that you showed us?
absolutely. Yeah. So these kinds of steps, you would need to apply some different applications. In this case, some of the applications we have can do some of these steps. Others are still being built out, and we can definitely. I'm trying to see if I have an example that I can show you here. Do I have one here? Well, so here's an example of a pipeline that we have, right? And this, I think if I click here, right, here we go. So this talks a little bit about one of these apps, right? This is a Biotia DX app that we have developed, and you can see that in the pre-processing step, we do clean the data, right? We do the pre-processing steps. So you would get fast QC data, you would get clean reads, you'd also get assembly, AMR detection, taxonomic classification. So these kinds of steps are available in the app. And these are the kinds of tools that we want to have available on GeoSeeq. So if there's anything that you would want that you don't see, you can let us know, and we can, try to develop it and make it possible. But this is an example of an app that would do what you're saying, right? So cleaning the reads
Right.
in the process.
Yes, I still have a lot of questions, but we have another the presenter here. But one
Oh,
thing is that the last one, sorry,
yeah, no worries.
the last, maybe we could also, if I'm sure that we have many questions, is it possible that we can just send email to you and then get some more
Absolutely.
questions?
Yeah, let me
Okay.
stop sharing and let me put my email in the chat. So, oops. IO. Did that go through? Yeah.
Yes.
So yes, please
Uh,
feel free to send me an email.
So the one thing is that actually it's publicly available, GeoSeeq, basically. Am I right?
Yes, it's available in the beta version at the moment. But we're continuing to build out this platform. And so yeah, it's
Mm-hmm.
available. And
Yes.
if you have anything that you want to see on there, don't hesitate to let us know so we can work on it.
Okay. So because some of my collaborators and attended in this workshop, actually they have a project separated from the collaboration work with me. So maybe they could use GeoSeeq for their own project. So as you mentioned,
Mhm.
they can just upload their file, the Pasteur file, and then run it and then do the analysis. And the last thing that I would like to ask you is that after the analysis, so when people publish the data, how do
Mhm.
we cite you?
That's a great question, and I don't have an answer. So let me figure that out and get back to you.
Okay, great. Okay. Thank you.
Okay.
I think that it's a very good first step I don't think it's enough, but we will keep asking you the questions.
Sure, that sounds good.
Okay, great. Thank you so much.
Yeah, thank you.
Thank you. And then
Okay.
please tell the niece that we are recording this section, so we'll send the file to her and you.
Sounds great. Sounds great. Okay, thank you.
Thank you. Thank you.
Yep.